I'm calling this video the anatomy of nitrogen metabolism. The idea is to dig into the basic moves that we make when we're doing metabolic pathways involving nitrogen. That will then set us up in the next two videos. We're going to tackle amino acid and nucleic acid metabolism specifically. So let's launch into the anatomy of nitrogen metabolism. In a past video, I talked about how nitrogen redox is a super complicated thing. If you write out all of the different oxidation states and you map out the pathways between them and the standard state reduction potentials, what you see is it's just an extraordinarily complicated mess. Some creatures deal with that horrible mess of chemistry, but we don't. We're with most creatures, and we leave nitrogen alone, and we leave ourselves basically in this ammonia state, this reduced nitrogen form state. As a biochemistry student, that's actually really helpful. Unless you're studying one of these weird creatures that does cool things, you don't have to worry about nitrogen redox. You can just zoom in and think about, well, what do we do with reduced ammonia type groups? That said, even boring animals do need to work with nitrogen, right? If we're going to build things like nucleic acids or amino acids, we have to do chemistry with these nitrogen. So the goal of this video is to ask two questions, which is what reactions do we do with nitrogen? And what are the molecules at the center of nitrogen metabolism? Now, this is not meant as an exhaustive video. I'm not going to go through all of the reactions we do with nitrogen. I'm just going to talk about the two most common. And I'm not going to talk about all of the ways that we move nitrogen around. Again, I'm going to focus on the most common. The idea is to get the core understanding of how nitrogen metabolism works in us, and then we can worry about adding details later. So, what reactions do we do with nitrogen? The primary reactions that we do is we shuffle around amines, right? We're not changing the redox state, which means we're moving around guys that look like this. Wow, that is terrible chemistry writing even for me. That's a 3+. NH, I'm just going to start over. NH3+. There we go. So we shuffle amines around. And doing so, there's really two core moves that we do. There are some others, but these are the two major ones. The first move is something called transamination. And what transamination involves is, say, take a glutamate, so the amino acid we know and love, an oxaloacetate, central carbon intermediate. And what I've done is I've circled or I've highlighted the amino terminus of the glutamate and this carbonyl on the oxaloacetate. What a transamination reaction does is it swaps these for each other. So it takes the amino group off of the glutamate and it sticks it on to that position in the oxaloacetate, and it takes the carbonyl off of the oxaloacetate and puts it where that same position in the glutamate. And what's cool about this is you might say glutamate and oxaloacetate, yeah, I'd seen those before. And you're like, oh, wait, alpha-ketoglutarate and aspartate. I've seen both of those before too. Central carbon metabolism and amino acids, they're actually really closely, intimately tied together. And one of the ways they're closely tied together is via this mechanism of transamination, which is when you take an amino group from an amino acid into a keto acid. This could be any amino acid to any keto acid. I just chose two examples here, or chose an example that's really common. But basically, anytime you take an amino group and you swap it with the carbonyl on a keto group, on a keto acid, you have a transamination reaction. So we're going to run into this a lot as we think about things like, well, how do we build nucleic acids? The second move that we make is a deamination and amination. This I'm showing a deamination reaction here for asparagine going to aspartate. And hopefully what you'll notice here is the difference is this group. So this amine pops off and gets replaced by a carboxylic acid. This process gives us a free ammonia that's floating around. This is an energetically favorable process, right? At standard state, the standard state reduction potential is very positive, means all else being equal, if we made the products and reactants all 1 molar, it would flow more towards products. We'd see a depletion of reactants, depletion of reactions, and increase in products. That's deamination. The amination move is thermodynamically unfavorable, but it's the exact reverse. So I've just drawn it as the exact reverse. We've got an asparagine and an aspartate. But to do so, every time we see this biologically, we're going to have to do some energy investment. So for example, there's an enzyme that takes the dephosphorylation of ATP into ADP and inorganic phosphate, couples that to this process, temporarily attaches the phosphate to the aspartate, and then lets the ammonia come in and do an attack. So that's the two moves. You take the nitrogen on and off. And so we saw transamination where you're moving an amino group from, say, an amino acid to a keto acid. This is just take the amino moiety on or off. And those two moves cover a huge fraction of the moves that we're going to see in nitrogen chemistry. There are a couple of others. There's other sorts of nitrogen transfer reactions, and there's variations on these. For example, there are things like oxidative amination and deamination, where there's a redox reaction involved. But overall, those are the two core reactions: transamination and the amination, deamination. Okay, what are the molecules that are at the center of nitrogen metabolism? So I thought it was interesting to look at it this way. So disappearing again. This is, if you go to Mayo Clinic Labs and just look up what are the reference concentrations for various molecules in human plasma, these are the results that come out. I just made a pie chart out of that. What you find is the biggest whack of stuff is urea, followed pretty closely by the amino acid glutamine and then the amino acid alanine. And so I've just shown glutamine and urea here. And then everything else, becomes much, much lower concentration. So if you think about it, we can sort of draw a line this way that says, whatever that is, 80-ish% of all the nitrogen in our blood, at a level of molecule level, not a nitrogen molar ratio, is 1, 2, 3, 4, 5, 6, 7, 8, 11, 12, 13. So 13 types of molecules make up most of the nitrogen, and half of it is glutamine and urea. So there's not, in a sense, nitrogen moving things around in the plasma is not that complicated, right? It's relatively simple compared to, say, some other biological processes we've run into. What's also interesting, though, is the distribution of things is actually quite different in cells. And so this is actually the whole metabolite component of cells. So this includes things like carbons and everything. But what's nutso is like 70-ish% of the molecules floating in your cell on a molar ratio are amino acids. So everything else, there's all these other molecules, ATPs and whatever, pyruvates floating around. 75% are amino acids. And that's, I think, pretty wild. So if you look at this, what I'm doing is I'm plotting these two next to each other. So this is plasma here on the left. This is cell on the right. And good time to pause and think about something. What do you notice that's different or the same? What do you see between these two? So I'm a big fan of pausing and thinking about it rather than just having me talk at you. So if you want, you can pause. Okay, you came back from your pause. What sorts of things do we notice? Well, a big difference is there's no urea, right, in the cell. The urea in the cell is tiny. I think it's actually on the list, but it's like down here somewhere. So there's way more urea in the blood than there is in a cell. We also notice that the dominant amino acids over here, if we just look at the top two, are alanine and glutamine. Whereas the top amino acids here is actually glutamate, which is a slightly different amino acid. And we've got glutamine here. But then alanine is all the way down here. We've moved aspartate up. So, if you look at it, there's been a shift in the distribution of amino acids from, say, alanine and glutamine to glutamate, and alanine. And what this reflects, these observations reflect, are what we do with nitrogen and how we move it around. So at a broad level, the way we transport nitrogen in the blood is as neutral amino acids. So we'll make something like glutamine and alanine and dump it into our bloodstream. That can be then imported by other cells that need nitrogen. So we're moving nitrogen that's sort of in our working stock around as neutral amino acids. But inside of our cell, the pool of usable nitrogen is actually stored in these negatively charged amino acids, things like glutamate and aspartate and glutamine. I mean, this is not an exclusive pool. But basically, when we need nitrogen to, say, build a base, a nucleic acid base, we're going to be grabbing amino acids and stealing the nitrogen from them to do that chemistry. And then finally, when we're done with nitrogen, when we no longer need it anymore, we transport waste nitrogen as urea. And so that's why urea is in our bloodstream. It's not in our cells. It's because it's in the process of being exported ultimately to be removed in the urine. Okay, so what this is hinting at is something that, at least, it's funny, I remember I was surprised by this when I learned this way back in days of yore, that amino acids act as more than protein components, right? That's sort of where I first learned about them, like, oh, amino acids, they like tryptophan. That's really important for building proteins. And that's true. But amino acids actually also act as a nitrogen currency, so moving nitrogen around. And then they're really important as metabolic intermediates. If you want to, say, build a uracil, you're going to be bringing in aspartate and you're going to be bringing in glutamine. So they're important for building other molecules. When we're thinking about how how amino acids get moved around, remember I said at one point that glutamine, the neutral amino acids, tend to be circulating in the blood. So if a cell needs nitrogen, it imports it using this cool transporter called the SNAP3 transporter. And this is a very, very clever transporter because what it does is it brings glutamine in indirectly doing this as a pump. And so the way that this works is this will come in, and it is a transporter that is a co-transporter that will also bring in sodium. So for every glutamine that goes in, a sodium ion comes in. So there's this came in and this came in. And what you'll notice is this is just based on the numbers from the Mayo Clinic and the paper that I pulled the numbers from for cellular concentrations. You're going from 1 millimolar glutamine outside to 17 millimolar glutamine inside. So you're pumping against the concentration gradient. How do we do that? Well, I said this is coupled or occurs with sodium being transported. And it turns out sodium is outside. It is at a higher concentration outside than inside because the sodium-potassium pump is continuously pumping sodium in and sodium out and potassium in, and this is using a ton of ATP to do it. And so basically, our cells invest energy to keep a gradient of sodium and potassium across the bilayer. And that allows our cells to do things like import glutamine by attaching it to sodium flowing in. So sodium and glutamine flow in together. The driving force is actually being provided by the sodium to bring the glutamine across its concentration gradient, right? So great. Now we've got our neutral amino acid inside of the cell. There's then another process that can happen where this becomes glutamate. So this actually there's a transformation. It's actually mitochondrial. It's a complicated pathway that I'm not going to go into right now for today. But ultimately what we do is we convert much of that glutamine into glutamate, that negatively charged amino acid that we're going to use as our internal nitrogen currency. Okay. Like I said, glutamate acts as a primary nitrogen currency. It's this high concentration thing in our cells. And why is it such a high concentration thing? Because it is directly adjacent to central carbon metabolism. So we've got our glutamate here, right? If we do a transamination reaction with oxaloacetate, that generates alpha-ketoglutarate and aspartate. Oops, my E rolled down. But what's cool about that, right, is we are basically exchanging between two central carbon intermediates, right? Oxaloacetate happens inside of the citric acid cycle at the top, sort of right before you get citrate in. Alpha-ketoglutarate happens down at the bottom after you've kicked off the two carbon dioxides from citrate. And so we're plugging directly into central carbon metabolism with glutamate and aspartate, right? So that's part of the reason that these are such common amino acids. I wanted to put a box in that, right? I'm just highlighting. Yes, these are central carbon metabolism or plugged into, a really critical central node in metabolism when we do glutamate and aspartate chemistry. Another interesting feature of these transamination reactions is that they are fast and have delta G's about 0. And that basically means that cells are continually balancing keto acid and amino acid concentration all by a Le Chatelier's principle. If you've seen my video on the non-oxidative pentose phosphate pathway, this is operating kind of the same way, that if glutamate concentrations go up, alpha-ketoglutarate concentrations go up and compensate to offset that, for example. And so cells can balance all of these together with a Le Chatelier's principle sort of approach. Okay, pretty much done covering what I wanted to look at. So we've talked about the basic moves, right? That's transamination and this amination, deamination. And then we've talked about what are the major players. And it turns out we're going to see a lot of amino acids, particularly glutamate, aspartate, and some glutamine in the metabolic pathways that we're going to see in the next couple of videos. Before I leave, though, I wanted to do one quick example where we just look at the start of one of these pathways and see if we can identify the things we've talked about. So We're going to be looking at the very start of pathways that is used to just generally break down amino acids. So we have some amino acid. Its identity doesn't matter because it's got an R on it. Excuse me. And what I'm going to do is put up the pathway showing the first couple of steps in its breakdown. So that's what I'm showing here. And this is a good chance to sort of practice looking at and understanding biochemical pathways. I'm going to ask you, what patterns do you recognize that are fit into the context of what we've already talked in this video? So what sorts of reactions, what sorts of molecules? So good chance to find the pause button and think about some patterns. So you're finding the pause button, you paused. Great, now you've unpaused, and let's talk about the patterns I see. So the first thing we see is at the top, this is a transamination reaction, right? We are taking a, let's figure out what this is, 1, 2, 3, 4, 5, that's an alpha-ketoglutarate, and we're swapping the amine off of our amino acid onto that alpha-ketoglutarate, making glutamate, and leaving now a new keto acid. So as we're breaking down general amino acid, a generic amino acid, one of the major steps is a simple transamination reaction. Pull the ammonia off of the amino terminus, stick it onto an alpha-ketoglutarate to make a glutamate. The next pattern that I see is this down here. We're seeing a deamination reaction. And this is actually one of these oxidative deamination reactions that involves an NAD plus and an NADH. But at the end of the day, the result is we kick off an ammonia. We have deaminated this. And incidentally, I'll just point it out because it's a cool part of the mechanism, is this we've now actually regenerated. So this is actually a cycle that we use to basically, you can think about this is taking the nitrogen, sticking it onto a common backbone, and then the enzyme that is going to do this deamination only needs to recognize glutamine, right? It doesn't have to recognize every amino acid. We just have to recognize glutamine to do this nitrogen chemistry. Okay, but overall, when we're getting into nitrogen metabolism in the next couple of videos, this is what I hope you'll be able to see. You're like, oh, there are transamination reactions, there are amino acids, there is deamination, and that'll help us as we understand how those pathways work. Okay, so that is what, oh, actually, I want to go back and do a quick summary, sorry. First, we use nitrogen in the reduced amine form. The two key chemical nitrogen moves that we make are this transamination and amination deamination. We see amino acids acting as nitrogen carriers in kind of two different roles. The first is in blood. We tend to see neutral amino acids that are just transporting nitrogen through the bloodstream, whereas in cells, we tend to have these negatively charged amino acids that act as nitrogen donors in transamination reactions, for example. And then that basically covers much of what we'll need to understand if we're going to understand things like amino acid synthesis. So look forward to talking about those topics with you. Until then, I hope you have a nice, I'm going to guess it's evening when you're watching this. So have a nice evening, and I will talk to you soon.